Good morning. Assalamu alaikum wa rahmatullah. Thanks for the invitations. I'll be talking actually about remissions, and Dr. Suzanne already introduced the topic very well. So before I go ahead and talk about the topic, let me get your feedback about the percentage of the patients who are going to achieve remissions in your clinical practice. So let's get the question. So what percentage of your patients are able to achieve clinical remissions? Can we start to vote? Yeah? Right, I think it's... So it's not bad. We're seeing actually almost 48% of the patients achieving clinical remissions. So let's get to the first slide here. Okay. All right, so let's look into the data assessing actually the remissions, sustained remissions in clinical practice. And you see this registry data actually showing, for example, in the percentage of the patients who achieved sustained remissions at any point during median follow-up of about 10.6 years. And you will see that using the DAS-28, almost 42% of the patients were able to achieve remissions. However, if you go into a more strict measure of remissions like the ACR-ULR Boolean, it's only like 17.5% were able to achieve clinical remission. So despite all the advancements in the field of rheumatology, we are still having more than 50% of the patients, or even more, not able to reach this important, basically, goal. Again, this is a real-world data, a remission to study looking into actually the disease activity trends over 5 years. And you will notice that, for example, from year 2009, represented by this orange bar, to the year 2014, represented by the light blue color bar, there is actually an annual remission rate increase from 36.7% up to 53.5% over time. Now, another thing which is important is that the moderate disease activity actually decreased from almost 33% down to 22%, and also there is reduction in the high disease activity from 11% down to 6.8% over the years, and this is really at the expense of using more biologics in this setting. Now, what about clinical remissions in the era of the biologics? And as already been mentioned, that there are certainly great improvement in the treatment of rheumatoid arthritis, and that's thanks to the basically the great understanding in the pathogenesis of rheumatoid arthritis. And as you are aware of the different steps in the pathogenesis of rheumatoid arthritis, the introduction of the cytokine inhibitors and the co-stimulation molecules like abatacept, rituximab, and also the other cytokines, these kind of molecules are working, in fact, outside the cell. And so they are trying to help prevent actually the activation of these kind of cytokines to the inflammatory process. However, now we have newer actually molecules which are working inside the cell to prevent actually the development of the pro-inflammatory mediators of inflammation. So in this basically study, which is looking into the combinations of methotrexate and etanercept, it's the COMET clinical study that was published in 2008. In fact, in patients who are methotrexate naive and have early rheumatoid arthritis, moderate to severe, you were able to achieve actually 50% of DAS-28 remissions if you use the combinations of etanercept and methotrexate, versus only using methotrexate, you end up having only 28% of the patients achieving remissions. So this is in the early patients with rheumatoid arthritis. How about those patients who have really long-term rheumatoid arthritis and they have high disease activity? The combinations of methotrexate and etanercept, for example, was able to achieve low disease activity in almost 44%, versus only 17% in those patients who are using methotrexate alone. Now, again, if you looking at the remission, we are able to achieve only 34% of the patients, the DAS28 remissions, versus only 13% on methotrexate. So this is really one of the advancements in the field, which is the addition of the biologics has helped in achieving more state of remissions than our traditional, conventional synthetic DMARDs. This study, which is the ARMADA study, looking into the utilizations of adalimumab, and you can see that the percentage of the patient achieving clinical remissions through DAS assessment in 6 months, 32% of the patients were able to achieve remissions, and if over 4 years, 43% of the patients were able to achieve remissions. If you look into this real-world data, looking from the Corona registry, looking into those patients who are over 8 years of follow-up, a significant number of patients treated with adalimumab achieved CDAI, low disease activity, and remissions. And you can see that over the years, there's actually an increase in the remissions. However, you have to take this data with a grain of salt, because you would notice that the number of the patients at the start of the therapy was 718, and at the end you ended having only 58 patients. And so there is some channeling bias here. Those patients who benefit from the drug without much of side effect, they will continue it. Those patients who are having certainly side effects or loss of efficacy, they may drop from the study, and that's what you see in here. Now, the clinical remissions in there of the JAK inhibitors, the JAK inhibitors, and as you know, the JAK family of molecules are really interesting, is that we have almost 4 members of these JAK inhibitors. We have JAK1, 2, and 3, and then we have the tyrosine kinase 2 inhibitors, the TYK2. These actually molecules are required for signaling the downstream of the cytokines and growth factors. And so each cytokine and growth factor receptor is associated with a pair of JAK family members. So we have actually 2 receptors, the cytokine 1 and 2 receptors, for the work of these JAK molecules. Certain cytokines and growth factors are implicated actually in the pathogenesis of rheumatoid arthritis. So you can see, for example, the interleukin-6 will work through the pair of JAK1 and JAK2, and also the interferon 2, which basically is associated with certain JAK combinations. And there are certainly other cytokines that are involved in the pathogenesis of rheumatoid arthritis that signal through the JAK kinase, as you can see in this graph here. However, the only homodimer among those JAK molecules is the JAK2. And in fact, we try not to target this one because it can be associated with worsening of the anemia and also the lymphopenia and other thrombocytopenia as well. So these, we have now 4 JAK inhibitors in the market. 2 of them were actually approved by the FDA for rheumatoid arthritis, which is tofacitinib and baricitinib. And tofacitinib was, proposed to be a JAK1 and JAK3 inhibitor. However, the FDA labeled it as a pan-JAK inhibitor. Baricitinib, for example, works on JAK1 and JAK2 pair. And the filgotinib and upadacitinib are thought to be, JAK1 selective inhibitor. Now, if you look into this pathway, how does the cytokine signaling leads to the activation of the pro-inflammatory cytokines and genes, you will find that binding of the cytokines to this, sole cytokine receptor will activate actually the JAK transformations, and then this activated JAK will phosphorylate actually the STAT, which will form a dimer that will translocate to the nucleus and then will lead to activate the transcription molecules and the pro-inflammatory factors. So there are certainly a number of these molecules can be inhibited, for example, by tofacitinib, which is a small molecule pan-JAK inhibitor. So it will target basically JAK1, JAK2, and JAK3, as you can see here. So it will block a good number of those basically JAK molecules. Now, baricitinib will inhibit JAK1 and JAK2, and you can see basically in the slides how it will execute its effect. And then the filgotinib and upadacitinib are supposedly JAK1 inhibitors. And you will see that the processes that can be involved in the pathogenesis, like lymphocyte proliferation and homeostasis, will be inhibited through this mechanism. JAK signaling, we try here to balance the efficacy and the side effects with the selectivity here. So if you look into JAK inhibition, there is a blocks actually the cytokine signaling, and including interleukin-6 and interferon, and therefore it may decrease the inflammation and protect the joints. There are certainly potential side effects to this, including the infections and hyperlipidemias. Again, if you inhibit JAK1, JAK3, there is really a tendency to decrease actually the NK cells and impair the protection from the infections and malignancy, and there are certainly side effects, including anemias and
Dr.
neutropenias
Zyad, we have
also.
5 minutes left.
Right. If you look into the selectivity versus specificity, if you have a non-selective molecule and it's supposed to target molecule 1, whatsoever the dose you are going to use, there will be affection of the other molecules that this drug will work on. However, if you have a molecule that's supposed to work on selective molecule and work on molecule 1, at a lower dose, it will work on this molecule. However, if you are using a higher dose, then it might affect the other molecules. And that's the problem with the JAK inhibitors. You will see that the dosage is really important, and it took the FDA a longer time to decide what dose should be approved, like in the setting of tofacitinib 5 milligrams BID and also the paricitinib 2 milligrams. Because if you are using higher dose, you will be able to target the other JAKs. So looking into upadacitinib, for example, the upadacitinib is 60 times more selective for JAK1 versus JAK2, and 130 times more selective for JAK1 versus JAK3, and 190 times more selective for JAK1 versus tyrosine kinase 2. If the drug is specific, there are certainly some differences between selectivity versus specificity. If the drug is specific, it means that it works only on that molecule that is supposed to work on, and even whatsoever the dose that you are going to use, it should not target the other molecules. So let's look into the clinical remissions in the era of JAK inhibitors in those patients who are methotrexate inadequate responses. In fact, this program is very extensive, and it covers a wide indications, including methotrexate inadequate responders, methotrexate naive, biologic inadequate responders. And so it's really extensive. I just talk about the methotrexate inadequate response RA patients. And you can see in this basically study the methotrexate inadequate responders patients. This is the oral standard assessing the efficacy of tofacitinib versus adalimumab and placebo. And you will notice that 18% of the patients were able to achieve remission, DAS28 CRP, like 18% of the patients achieved remissions in the tofacitinib arm versus 16% in the adalimumab. And you can see that it's really statistically significant against placebo, but as good as adalimumab in this setting. The paricitinib, when we're talking about actually remissions, again, you will find that the paricitinib achieved 24% remissions in the setting of this study, which is the RA BEAM, and 19% of the patients in the adalimumab group achieved remissions. So this is study also showing that the paricitinib is statistically significant against placebo. However, it did not reach statistical significance against adalimumab. We are talking about the remission state here. Again, if you look into this upadacitinib select compare clinical trial, you will find that the remission was achieved in 29% of the patients in the upadacitinib group versus only 18% in the adalimumab. And you will see that it is statistically significant against adalimumab and placebo. Right. So in summary, achieving clinical remission is becoming increasingly important at the era in the RA treatment paradigm. And we have noticed that the introduction of the JAK inhibitors have raised the bar for this situation. The JAK inhibitor selectivity may lead to an improved safety profile or the ability to achieve exposures associated with peak efficacy without compromising the safety. And consequently, the selectivity has the potential to permit therapies to achieve an optimal benefit-risk profile. So with this, I end my talk. Yeah. Thanks a lot.
Thanks a lot. Thank you for this excellent presentation.